Yeah, hi everyone. Am I audible?
Hi, yes, you are audible.
Okay, perfect. Yeah, so before starting the session, could you please introduce yourself, both of you, so I can know your background? So,
Um,
I'm not sure. I guess I'm not able to hear you. So, is there some issue? Yeah, still I can't hear you. So, is it the case for me or is it for you, Akansha?
oh, yeah, I also can't hear her.
Okay. Yeah.
It is some issue at her end.
Yeah, could you please try to rejoin? Maybe it will resolve your mic issue. Yeah, no issue. You can also update in the chat box. I just want to know your background, what exactly you're doing currently. Still, I can't hear you.
Meanwhile, can I go for
Yes,
myself? Yeah.
Yeah, yeah.
yes,
Now she is here. Yeah, we can hear you, Kansha.
Okay, okay.
yes. So yeah, please go ahead.
Yes, ma'am, you can go ahead.
Okay, thank you.
Yeah.
So I'm a fourth year PhD scholar in ICGB New Delhi. So I am working on a tuberculosis where I'm trying to understand the how intercellular pathogens affect the T cell immunity. That too in terms of the epigenetics and metabolism.
Okay.
Hello, am I audible now?
Yes.
Okay, so my name is Akanshi Sharma, and I'm currently enrolled in a PhD program in CSIR Impact Tourism. And I'm first year PhD scholar. So I've just recently enrolled in August. And basically, my project will it's related to microbiota. So I'll be involved in transcriptome analysis and all that. So that is why I've joined this program.
Okay. And yeah, sure. Thank you for the introduction. And just want to confirm two things. So you guys have any understanding or knowledge of, let's say, for example, Linux command line shell scripting and the coding language?
So I have no experience in coding language and yeah, so nothing related to the bioinformatics. I just have done the proteomics analysis that too using the online software tools.
Okay. And what about others?
No, even I don't have any background.
Okay, sure. Because we are going to write the code part. So I just want to know, like, I have to explain from the basic part or I have to explain at an intermediate level. So yeah, thanks for the confirmation.
Yes.
So let me share my screen. Yeah, so can you see my screen?
Yes, yes.
Okay, perfect.
Yes.
Okay, perfect. So today we are going to start with the basic session, that is the introduction part. And then I'll take a couple of session on Linux command line and for the coding part, I'm not going to take any session on coding part, but whatever the code I'm going to write, I'll explain from the basic part. What exactly the code I'm going to write, what exactly the parameter I'm using, how exactly at this level, okay, this is the code, why I'm using this code. So I'll explain those parts. But if you want to have your understanding or you want to know, so I would suggest to follow any lectures. You can take any. So there are a lot of resources on GitHub on Google for R and Python basic. You can also refer YouTube, and from there you can collect the basic knowledge like what is the variable, what is the data types, how we can write the print statement, how we can write the logics or for loops. This part is the basic coding part that you can try to read, and then rest of the thing I can explain. And if you're not able to find any resources, then let me know. I can provide the resources, but these resources are easily available. So you can easily search on Google or YouTube part.
Sure.
So starting with the NGS part. So NGS stands for next generation sequencing. So if you see the central dogma for molecular biology, so this is the structure we follow. So we have a DNA, and after transcription, basically we have the RNA, and from the RNA, basically we have the translation, then we will have the protein, and we are doing the study on proteins based on their phenotype expression profile, and that study called as a cellular phenotype. So this is the entire dogma. If we try to see the same structure in the field of genomics, so whenever we are doing the study for DNA, that comes under the field of genomics. Whenever we are doing the study for transcript, that is transcriptomic analysis or gene expression profile, that is related to RNA. So we call that particular domain thing is transcriptome. Same thing, study of protein comes under the category of proteome. And study of cellular phenotype or expression profile, that comes under the cellular phenotype category. So this is the central dogma for the genomics part. Now, why we use NGS nowadays? Because let's say what exactly we are doing. So the main thing is we have a DNA and we don't know what exactly the sequence that is present on DNA. So we have to find the sequence that is present on unknown DNA. Okay. So that is the task. So as of now, we are using NGS, but the previous or traditional method was we try to take the DNA fragments and try to find the sequence. But that is time consuming. Means if I have to find the entire whole genome sequence, so I need to do a lot of fragment and I need to pick one fragment, take this fragment, find the sequence, then need to go for next fragment, then need to go for next fragment. So this is time consuming, and let's say you have to pay a lot of money for doing the entire whole genome sequencing. Now, what is the advantage of NGS? This is massive parallel sequencing. So there are two terms, like we are generating high throughput data, means a lot of data. Second part is we are doing massive parallel sequencing. It means whatever the fragment that I have, I can find the sequence all together. Basically, we are finding the sequence for each fragment parallelly. So that's why it is called as massive parallel sequencing. So we can have different lengths of DNA or RNA. For that, we need to find the sequence. And these things are running in parallel. So basically, we are generating a lot of data. So we can do the entire whole genome sequencing within a short period of time. So this is the advantage. One advantage is we are generating a lot of data, and basically we can do the sequencing within a short period of time. So this is the advantage of NGS. Now, if you see the next part is that is the evolution of sequencing, means what are the different generation of sequencing that we have. And for example, if I have
Sir,
to
can
design,
I ask one question?
yes,
So I wanted to ask, means we are doing the sequence in a one go, like we are not generating fragments here.
no, we are generating fragments because let's say if this is a sequencer, so each sequencer has a capacity. It means it can take a read up to size 150 base pair or 200 base pair, 250 base pair.
Hmm.
But if you see the entire genome, it can be more than that. So you have
Yes.
to do the
Fragment.
fragmentation so that it can
Okay.
fit into the sequencer
Okay.
requirement. So that is the case. Yeah, that
But
part we will discuss. Yes. Yeah.
okay, then how it is different?
Different from what?
Different from other techniques that take that are time consuming, like different other
So,
sequencing
so
techniques.
let's say, for example, in this case, what is happening is in the normal traditional method, we took one fragment and find the
Okay.
sequence. But
Yes.
in the NGS part, we will have a base, let's say surface, and basically we have a lot of spike. So we try to capture all the fragment. So basically we are trying to capture all the fragments and finding the sequence in a single go. Means we don't have to do one by one.
Okay. Okay.
In the traditional approach, you cannot go to the next part unless you solve the first one.
Mm-hmm.
So
Okay.
that's the time consuming part.
Hmm.
Now, yeah, so we have a lot of generations. So first generation, second generation, third generation. So first generation, we also call it as a traditional approach or traditional method.
Triangle sequencing.
And
Hmm.
within that, we have a Sanger sequencing and Maxim Gilbert sequencing. And
Hmm.
I hope you are aware that is they are using the logic of DD antiphase, basically chain termination method. But the one thing you need to remember is in this particular process, we are dealing with a fragment size that is 500 to 1K base pair fragment. And the second part is this process is slow and very expensive. And the logic we are using to find the sequence is DDNTPs, that is basically used for 10 terminations so that we have a lot of reads and we can run on jail to find the sequence. When you try to compare the first generation with second generation of sequencing, so what is happening here is in second generation, we have a lot of instrument or lots of sequencer. Okay, that is falling in the second generation. We have the Illumina, we have Iron Torrent, we have Solexa, Roche. So these are the different sequencing platform that we have in the second category of second generation. So the first generation and second generation is dealing the short read sequencing. Means whenever you have any requirement to find the sequence of short read, you can use either first generation or second generation. That is the first thing. Second thing is in the second generation that is also called as NGS that we have seen in the first slide. What is the advantage? High throughput means generating a lot of data and using parallel sequencing concept. Giving high accuracy means the error rate will be very less in this particular data or in this particular result. And basically you have high accuracy. The size of the fragment you can handle is 50 to 500 means basically also you are handling the short read fragments to find the sequence. And this is the faster and more affordable thing. Why I'm saying high accuracy is when you have a lot of copies for a single read. So you may be aware of that this is the reference part and we try to map our reads. That is the process of alignment. Okay, so we have a lot of reads. So let's say at this particular position, in this particular section, we have A. In this particular section, we have A. Then in this also we have A. And let's say for rest of 2, I have T. So basically, I have the probability 3 by 5 means it is providing me support, or you can say, let's say, giving me confidence to say at this particular position, we will have A. So that's why we are generating a lot of reads that is going to map to the same reason so that you may have know about the concept of consensus sequence. So basically, we try to see what exactly the probability or which particular nucleotide is present in more quantity at this particular location so that we can figure out at this particular position what will be the nucleotide that is called by the sequencer. So that's why we have a high accuracy part. One thing is both the method is handling the short read sequencing. The comparison between first and generation is this is slow and this is basically faster process, and you have to pay less money or spend less money in the second generation compared with the first generation. Now we have the last generation, that is third generation. And within that, we have a two sequencer, that is Nanopore and the PAC5. Both the sequencer is basically handling the long reads. So what is the long read? Basically, we have a longer fragment DNA and we want to find the sequence in a single go. So when we have to use third generation, there might be question, okay, why I can't use third generation, why I can't use second generation? So basically, depending upon your question requirement or projects, let's say you need to find the long indel variants. So that time you cannot use the second generation because that is handling it short range. And if you have to find long indel variant, structure variant, for that kind of question, you have to go for the third generation, where you are going to find the sequence for long range. So that's why we have a different questions and we can use different sequencing platform to solve it. One of the disadvantages is this method is having a lot of errors. So basically this process is going to provide lower accuracy than the NGS method. But the advantage is you can do the sequencing for longer fragments. So this is the entire evolution of sequencing. Moving to the next slide. Now we are going to see the entire workflow for the NGS, like what exactly we are doing in the entire workflow. Okay. So this entire workflow is divided into 4 steps. One is the sample collection, then the second step is library preparation, third is sequencing, and the fourth part where we generally jump into, that is data analysis part. So what exactly we have to do is in the sample collection part, whatever the sample you are handling, it basically means, for example, whatever the model you are handling, for example, human model, mouse, any sample you are handling, you need to collect the genetic information. And depending upon the pipeline you are going to run at the end, you have to collect the genetic information. Either it can be DNA or RNA. Let's say, for example, I need to do the analysis of, let's say, variant calling. So for that purpose, I cannot isolate RNA because I have to do variant calling. So I'm going to isolate the DNA part. Same thing if I have to do the gene expression profiling study. For that purpose, I'm going to isolate the RNA because RNA is going to give an idea how many transcript that is produced by that particular gene. That will give the expression profile. So this is the step one that we are
So
trying.
sorry, can you just repeat it? Like when we can use RNA and when can use
So,
DNA?
so DNA, when you have a thing that is related to DNA, for example, when you're going for DNA-seq analysis, that's a variant calling, ChIP-seq, ATAC-seq, where DNA is involved. For example, if you want to see, let's say, where exactly the mutation is present, for that time, you need to collect the DNA part. RNA part means when we study the transcriptome profile, that is gene level. For that
Okay.
part, you have to isolate the RNA. And let's say, for example, from the same sample, you want to perform variant analysis and also the gene expression analysis. That time you have to isolate both DNA, RNA. So
Got
depending upon the analysis and requirement, you're going to isolate the genetic information. That is the one thing. Second thing is, as I was saying, the sample types can vary. For example, we have a lot of analysis, a lot of assay. So we have, let's say, single cell analysis, transcriptome analysis, whole genome analysis, exome sequencing, target sequencing. So depending upon that, sample types can vary. So this is the step one part. After having the sample collection, you need to go for library preparation. So this is the main step. So whatever we are discussing, this is the wet lab part. So I'm just giving the basic intro. If you want to know more about, you can read it. There are a lot of literature paper that is there. From there, you can collect the entire information. Now the step two is basically library preparation. So that is clear is I need to send my sample for sequencing. And whatever the sequencer you're going to use, they have the criteria, means they have the specific length they can handle. So you have collected the bigger chunk of template DNA. Let's say the size of this DNA is 2 kb. So this DNA is not going to fit in the requirement of the sequencer if I'm going to use, let's say, Illumina. So Illumina is basically short read sequencing that can handle, let's say, 150 to 250 base pairs. I'm just giving a dummy example. So this 2 kb is not going to fit into the requirement. So what I have to do is I need to do the fragmentation, means I have to create a smaller fragment. So there are a couple of processes. You can go for sonication or enzymatic assay to create the fragmented DNA. So what we have is from the bigger chunk of DNA, we have created fragmented DNA, that is smaller length DNA. Then the next process is you need to add adapter. So adapter is
I
nothing
have a
but
question here.
yes.
So how do you make sure that the reads that you are getting from this library preparation is between that range? Like how do you decide the method of this fragmentation?
So I'm not sure about the parameter they're using while doing the sonication. So there is within the sonication, how much pressure or force you want to apply. So that part I'm not aware how exactly they are doing. But the thing is, or let's say for example, if you're going for enzymatic assay, so up to which length? So it's not like that. I can have a length of 150 base per always. You will have a multiple
Right.
fragment size. You will have 130, 150, 160, 170.
Mm-hmm.
But I'm not aware how exactly they are designing or while doing the sonication, what parameters they are including so that they can have a fragment size less than 200, let's say, for example.
So there are no chances that it will be beyond 500 base pair, right?
No. So they will
Okay.
try to have an experiment such that the length will be in the format they are expecting or they want to create.
All right. All right. Okay. Thank you.
Yes. Now let's say we have the fragmented DNA. Now we need to add the adapter. So adapter is nothing just as a DNA sequence, small stretch of DNA sequence. So the basic logic of adapter is when you are going to use Illumina, Illumina has a flow cell and this flow cell has a lane. So let's say this is a lane. And if you zoom in the lane surface, so you will see a surface which has a lot of spikes. And these spikes are nothing but the complementary sequence of the adapter that you're going to use. Okay. So let's say if this adapter is having a sequence of triple AG, so this will be having triple TC, basically the complementary sequence. Why we are having this? Because I want to capture the read. So that's why we have the adapters. We want to ligate the adapter from both the end. So now if you see the product, final product, we have the read that is in the green part. We have the adapter that is at the end part. And if I'm going to load the sample in this particular area, let's say for example, in this particular well, so what exactly I'm trying to do? This is the adapter, which is complementary sequence. They're going to hybridize with the complementary sequence. So basically the role of adapter is I want to capture the fragment DNA so I can make the copies. So this is the main role.
Sir, I have a question.
Yes.
So by which force the adapter is ligating to that particular DNA?
So
Like
basically,
how?
you're going to ligation. You're going to use DNA ligase enzyme. You're going to use. They are going to and or you're going to stick the both the end whatever the read fragments you have and this end-to-end ligation they're going to do.
How the adapters are made here?
So this is just as a DNA sequence. For example, you can design. For example, how you're designing the primer. Let's say, for
Okay,
example,
okay,
say.
like that, fine.
So this is a DNA sequence that you can decide. Okay, this is the stretch I want. How
Hmm.
many base pair I want. So this is the base pair you can decide, and same base pair will go there as a complementary.
So why the adapter has two colors here? What is the significance?
No, this is just as a coloring pattern, so it can see distinct between the green part. That's it.
Okay, fine.
Now you have the adapters, you have the complementary sequence. So basically, it can hold the adapter part and basically capture the fragmented DNA. So now I can add other chemicals, other DNTPs. So basically, chain elongation will happen. So I can have the copies for that particular template DNA. So this is the process I want. So that's why we try to have the sequence library where we have the read and we try to have the adapter. Now there is one more concept, that is the concept of barcode. So let's say nowadays sequencing is costly. And let's say I don't have too much fun to waste money. So let's say I'm running a company and I can provide the sequencing. And let's say you two are coming, hey, could you please sequence my sample? So let's say you are providing sample A. So let's say one person has given sample A and one person has given sample B. Okay. Now this is the flow cell area, and in the flow cell we have the lane. Let's say, for example, this is lane A, let's say. Or I can say give a different name, let's say to make it different, ABC. So this is a well where I can load the sample or where I can load the library that I have prepared. So let's say, for example, the requirement is for A, I need to generate 50 GB data. Let's say, for example, I'm just giving a dummy example to explain what is the concept of multiplexing. This one, I need to generate 50 GB data. For this one, I need to generate 70 GB data. And at this well, I can generate maximum 150 GB data. Okay. So I cannot do, for example, this way is I'm going to add sample A in this lane and sample B in this lane. Because basically I'm wasting the area or the space of generating the data. Because in this case, I'm losing, for example, 80 GB data information. In this case, I'm wasting around 100 GB space data information. Because if I'm going to use the well, I cannot use again. So what I'll use it, I'll try to combine it. So that is the concept of multiplexing. Means you are going to pool the sample. After pooling the samples, I can add in the same well and I can go for sequencing and I can generate the data. But the next question is how you can identify which DNA or RNA coming from sample A or sample B. So for that particular part, we are using the logic of barcode. It means I have a sample B. I'll do this entire process while adding the adapters. This is the adapter. I also add within the adapter, there is a stretch of sequence that we call as a barcode. That is a unique ID, like Aadhaar card. For example, if I have to identify someone else, so we have unique ID that is our Aadhaar card. Same thing, whatever the transcript or DNA that is coming from sample B, they will have this tag or they will have this sequence that is unique to sample B. Then we will have our reads and then we have the next adapter. Same thing, we are going to do it for sample A. So sample A will have different barcode ID, sample B will have different barcode ID. And from this barcode ID, later we can go for demultiplexing, means we have to segregate the sample that is coming from sample A and coming from sample B. So that is the concept of demultiplexing. Multiplexing means pool the sample. Demultiplexing means separate the sample data. Because I have to, if you have shared the samples for sequencing, I need to provide the data. So I cannot provide sample B data along with your data because I have to segregate first so that I can provide only to you sample A data and I have to provide sample B to sample B person. So this is the concept of demultiplexing. Is it clear?
Yeah, I have a couple of questions.
Yes.
So in case of this barcode, let's say in like for example A, you have given that I have 1000 copies of the reads, and
Yes.
in the other set of the data you have, let's say 2000 copies.
Okay.
So the barcode that you are using will be single, like single unique code, right? For all the
Correct.
1000 copies in one case, and that,
Correct.
right, it will be a single barcode.
Correct.
So this thing is applicable to Illumina.
It is applicable to any platform where you're going for pooling.
Okay. I'm I'm confused between this 10x genomics and this Illumina. Can you please just help me with that?
So 10x Genomics is basically doing single cell. So they have a single cell, so they will design the barcode for each cell. In this
Each
case, we are
cell.
designing the sample. So we have one sample, but if you go
Mhm.
for 10x platform, we have many cells. In this case, we are
Yeah.
handling the tissue level. So we have one sample. In this case,
Mhm.
we are rupturing the tissue and isolating all the cells. So we have many cells act as an individual thing. So you have to create a unique tag for each cell. So if you have 1,000 cells, so you are going to create 1,000 unique barcode ID for each cell so that I can track which transcript is coming from which cell.
So how do you get to know that this barcode has been given to this cell type? So
So
that's
with
not
that is so no, that is the concept of flow cytometry. So let's say, for example, if you are not aware of this part, so let's say I'm just giving this simple structure.
Mhm.
In this particular lane, I'm going to pass single cell. Okay. In,
Absolutely.
from this cell, we have the wild droplet.
Mm.
And when we are going to merge it, so we will have a scenario, a bigger wild droplet that is going to capture single cell, and we will have the beads and other chemicals that we need. Then we are going to rupture this cell, the transcript will come out. We are going to capture these reads or transcript onto the bead surface, the same way like we are doing on the flow cell. So bead is basically inert, which has the spike. That is the complementary spike of the adapter.
Okay,
So
so that
whatever,
beat will capture all the transcripts.
correct.
Okay.
So
Understood.
this is happening in each cell.
Okay. Thank you.
Sir, firstly, you were saying that barcode is specific for each DNA fragment of a
No, no, not
particular.
no, not DNA fragment. I said for sample, sample A and sample B. I said
Okay. Each means that it be whatever fragment it is, but it will have a specific barcode.
correct. Let's say, for example, from sample B, I have 5,000 fragment. All the 5,000 fragment will have same barcode.
Same. Okay.
For
Fine.
sample A, if I have only two reads, two read will have the same barcode. That is different from sample B barcode. That's it. Any other question?
Um, no.
No.
Okay.
Yeah.
Now we have one more process, that is the amplification within the library application. Amplification is basically deciding the amount of data you want to generate. Okay. So basically we use the PCR, that is the common process, to increase the amount of DNA, that is polymerase chain reaction. So it is totally depends on the coverage. So for example, for each of the assay or for each of the experiment, we require different coverage. Let's say 50x coverage, 100x coverage kind of thing. So depending upon that part, we can go for amplification. And if you're not aware of the coverage concepts, there is a simple concept. Let's say this is the reference path, and let's say this is a reason. If I'm saying I'm going for sequencing with 10x coverage, it means on an average there will be 10 reads that is going to support this particular reason. So there will be 10 reads. If I'm saying 50x coverage, there will be on an average 50x coverage. So for example, in a couple of places, you will have 45 read that is supporting the reason. You may have 40 reads, but you're going to do the main calculation. You are going to have on an average 50 reads. So that is the concept. If you're going to have 100x coverage, you will have on an average 100 reads that is mapping to that particular reason. So this is the concept of sequencing depth or the coverage.
Sir, I didn't understood the amplification part. Can you just repeat it?
So I said, for example, amplification means I want to make more data, more copies.
Okay.
So PCR is basically doing the cycle and we generate a similar copies. So that is the
Mm-hmm.
amplification.
Okay.
So that process, for example, you want to make many copies. So you can go for PCR and then you can increase the amount of DNA. So that is the process of amplification.
Uh-huh. Then what you said?
I was saying the explain concept of coverage part. What is the meaning of coverage? Sometimes you may have seen 10x coverage, 50x coverage, 100x coverage. So
No.
coverage means how many reads that is supporting that particular reason or how many reads that is overlapping to that reason. That is the coverage part.
So more the coverage, more will be the confidence in the data. Is that the thing?
And no, more the coverage means you have a more read that is supporting. Depending upon, for example, if I am looking for variant calling, so let's say this is the reference part, and let's say for this reason, there is a two reads that is supporting for
Hmm.
this variant. And for this variant, I have 50 reads.
Hmm.
So obviously, I will have more confidence to say this variant is correct instead of this
Hmm.
part, because
Right.
these two read may be incorrect. So these may saying, okay, at this particular location, you have a variant. But if you're going to have more coverage or more read that is going to overlap, having the variant or do not have the variant, that can verify whether this is correct or not.
Uh, we already have a reference there, right? Like that we already have.
Correct. In the reference part, you have a G, but when you are overlapping your query data, it is saying at this location you have A. It means you have mutation.
Okay.
But if you have more read that is saying at this location you have mutation that is A, that is providing more confidence score.
So is it possible if you can just explain this with an example, any example of biology?
So yeah, it's the same example I'm explaining. For example, in the reference part, you have G, and in
Mm-hmm.
this particular part, you have T. Now you're going to map your read, and in your read part at the same location, you have A instead of G. It means ideally it should have G at this location, but in your genome, it is changed to A. Means you have SNP, single nucleotide mutation.
Mm. Okay.
Same thing is happening at this location, but what you can say is I have a confidence score that is high at this location. Means a lot of read that is mapping to this region, which is saying you don't have T, you have A. But at this location, you have only two read, that is very less coverage. Means that can be a false mutation also. So basically, this is the concept of probability. So let's say, for example, in a class, if 10% is saying you are bad, and if, for example, for other person, there are only 2% who is saying he is bad or he is bad. So you can say, okay, the 2% may can say lies. Let's, for example, they may be, let's say, because of something they have some, let's for example, their bonding is not so good. So that's why they're saying C is bad or he is bad. But if we have a more number of students who is saying C or he is bad, it means that the person is not good. Means he has done something wrong. So the same logic we are applying here, more number of read that is supporting, we have more confidence score or more accuracy we are adding in the result part.
So, out of curiosity, I have a question
Yeah.
that may sound silly to you, but is there any chance that you miss some of the reads that cannot be mapped to the reference genome?
Correct. If they are not passing the quality filter. So because let's say
No.
that when you're going for alignment, a lot of parameter is there
Yeah.
to decide which reads you want to keep, which reads you want to filter them out. So a lot of quality control is there.
So, do you like get into that losing the reads?
So you cannot use all the data because some data, let's say for example, I can give a simple example. Let's say
Mm-hmm.
I can give an example of single cell. Okay, let's say I have one single cell condition, and this is the second single cell. In
Mm-hmm.
this wild droplet, I captured one cell. In this wild droplet, I captured two cells. So now let's say whatever the transcript you are going to collect, that will provide the gene expression profile, means the activity of the cell,
Okay.
whether
Yeah.
the cell is producing that particular transcript for that particular gene or not. So we will have the expression profile. Okay. Now let's say I'm tracking one gene, let's say TP53, and I got 50 transcript that is mapping to TP53 in cell 1. Okay.
Hmm.
But in this case, you are not aware this particular droplet captured two cells because you cannot see within the droplet part.
Hmm.
Now let's say when you're rupturing these two cells, you have the number of transcript that is there is 150.
Okay.
So now you're going to say this particular cell has very high expression profile for TP53 compared with this cell. Correct?
Hmm. Hmm.
But you are doing a mistake because this is the doublet condition.
Right.
Because the content you are collecting, because it is the combination of two cells.
Hmm.
So for doing any kind of analysis or doing biological interpretation, you have to do QC. So you have to filter these kind of cases before comparing the cells. So the same thing we do the QC when we do alignment, when do any kind of analysis. So there is a step that is quality control. And within quality control, there is a lot of parameters.
Okay. So, okay. Okay. Understood. So there may be the possibility that you are not able to get the all the data that we are expecting. For example, if I was expecting, let's say 5,000 genes were supposed to be found in my data, but because of the limitation, or you can say anything about this alignment, I am just getting 3,000 reads. So
Correct.
that's
For example,
possible,
yes,
right?
for this gene, let's say there are a lot of read that can map, but because of the quality control, they did not pass.
Right,
So we are not having any coverage at this location. Means there is no read that is overlapping or
right.
mapping this region. So that's why
So we
you
lose
will
that
have a
information.
zero value.
Okay, okay, okay. Thank you.
Yeah. So this is the part of amplification. Then the next part is basically the sequencing. So this is simple. Now I have the sample. I need to send the samples to sequencing platform and they can find the sequence. That's it. But there are a couple of sequencers because let's say there are different platforms like Illumina, then Roche, Nanopore, PacBio. So all of the platform cannot use the same biology because they have the patent something. So each sequencing platform is using different sequencing method to find the sequence. The end product will be the same. You will have the sequence, but the way to find the sequence is different. So I can give one example is let's say Illumina is using the concept of fluorophore. Means, for example, this is the template DNA. Let's say this is the primer. Then we are going to add DNTP. So let's say in the reference part, I have 80. So what we are going to do is we are going to add one DNTPs at a time. So let's say, for example, I'm going to add DGTP. So G is not complementary of A, so we will not have any signal. So what exactly the signal means? The DNTPs that we are going to add that will have the fluorophore tag. Okay, so basically I'm going to add a tag. So whenever it is going to hybridize, it is going to produce a signal, and that signal we are going to capture. Now in the second round, I'm going to add C. C is not complementary of A. We are not having any signal. When I add DTP, so basically T is complementary of A. T is getting added and it will produce the signal. I got the signal. I capture the signal. I got to know in the reference part there is A. So this is the way they are trying to find the sequence. Same thing, someone is using a different concept to find the sequence. So the biology may be different, but the ultimate goal is to find the sequence that is present on the DNA tag. So this is the concept of sequencing. So you are going to read the DNA sequence that is present within the sample. And we have a lot of sequencing platform. And you can select the sequencing platform depending upon your requirement and analysis part.
Sir, I have one doubt.
Yes,
Since we already have a reference, so we know the sequence of that reference, correct?
correct.
And the fragments are generated and fragments are aligning to that reference. So automatically we know the sequence of that fragment. So why we are sequencing then?
No, how you're going to do the mapping unless you don't know the sequence of the fragment. For example, let's say I'm giving this is the reference and we have a let's say T and T, and you have two reads. You have a read, let's say this is the read. So how
Hmm.
you're going to decide this read is going to map at this location or this location? Because let's say you are going to have the information. For example, this is AA. So this will map to this region. This will have TT, and this is going to map this region.
So I know it means I know it because of the reference that you were saying that there is a reference.
No, for example, I'm going to take your genome. So how you can know like what is the sequence that is present in your genome? Reference is because you're comparing with the reference, so you can find out the sequence that is present in your genome. So reference is like a base. For example, if I have to find is there any mutation, or let's say in future I have a disease, or I may have some disease, I want to do
Hmm.
some testing. So I'm going to do my sequencing. And let's say whatever the sequence I have that may be not similar to you. You may have, let's say, different sequence for any particular gene. I may have a different sequence for that particular gene. So if I have to do that part, I'm going to do the sequencing. Then I am going to find the sequence of the reads, and that is going to overlap because we are going to do the mapping part. So we should know the sequence that is present on the read, their quality score and everything. That is a part of alignment. You cannot have a known read and you go to do the mapping. So that is the concept.
Hmm. Okay.
So the next thing is, yeah, so this is the sequencing part. And the last step is the data analysis part. that we are dealing. So basically, whatever the data we are getting from the sequencing part that we have to handle, that is the completion analysis. So generally, all the sequencing platform is going to produce the NGS data in the format of FASTQ. And this FASTQ will contain the information that is basically, it will contain the read information. For example, in your sample, if you have 4 reads, so this FASTQ will contain the information for 4 reads. So basically, FASTQ file is a kind of 4-line information. It will have the header part, then it will have the sequence part, plus operator, and then they have the quality score. That is the Fred quality. I'll explain in details what is the Fred quality in upcoming classes, but as of now, you can just remember FASTQ file has a 4-line of record for each read. So basically, it has the header part and the sequence part and the quality score also. And from this point, you are going for different pipeline. As I was saying, if you are going for transcriptome analysis, you have to follow different pipeline. If you are going for variant calling, you have to follow different pipeline. If you're going for chip-seq analysis, you have to follow different pipeline. If you're going for ATAC-seq, you have to follow different pipeline. So for each kind of analysis, you have to follow different pipeline. But the starting raw data will be the same, that is the FASTQ file. From the FASTQ file, you're going for the alignment to generate the align reads, that is in the format of either BAM, SAM, or FRAM. And from there, you can have the variant calling for the variant analysis. You can have the quantification for the transcriptome analysis. You can have the peak calling for chip-seq assay. So depending upon the analysis, you have to take the direction. That's it. So this is the step 4 where we are handling the analysis step. But the starting data will be always the same. That is the FASTQ for each sample we have. Now moving forward, so these are the applications. So NGS is used nowadays in all the domains. So where you are going for whole genome sequencing, we are depending on NGS. Whether you're going for target region sequencing, whether you're going for gene expression profiling, we are generating the data using NGS. Whether you're going for single cell analysis, that is cellular heterogeneity finding, you're going to use NGS. Whether you're going to do the chip-seq analysis, DNA protein interaction, also we are going to use NGS. Even you're going for metagenomic study, means whenever you are dealing with any environment sample, either water sample, soil sample, gut microbiome, means basically you're targeting to find the microbial diversity. That study comes under metagenomics. That time also we tried to generate the data using NGS, means we are generating a high throughput data, a lot of data using massive parallel sequencing. In this case, we don't have DNA. Basically, we tried to use 16S ribosomal RNA. That is the concept of metagenomics to find the microbial diversity. So that is a different domain. But as of now, just need to note down that in the each of the field, NGS is used to generate the data. And the data we will have is the FASTQ file at the end. Now I'm going to cover 2 more slides. One of the slide when we are going to have the gene expression data, because we are going to do this workshop on RNA-seq analysis. So RNA-seq analysis, basically we are having gene expression data. Basically we are doing transcriptome study. So the first experiment that is the microarray. Then we have the RNA-seq. Then we have the single cell and other kind of transcriptome study. So in the microarray, what we are doing is we have the healthy cell condition, we have the pathological cell. So basically we are trying to do comparative analysis. So we are trying to compare 2 conditions, normal and let's say treated condition. You're going to culture the cells, you're going for the isolation, and then you're going for reverse transcription to create the DNA. And you also added a tag. Okay, so now each transcript will have that, or each template DNA will have the tag. So in the microarray part, what we are doing is we have to, before starting the experiment, you have to design the probe, means for which gene you want to see the expression profile, means for which gene you want to compare healthy versus pathological cell. So you have to prepare. So for example, I'm going for 10 gene study. So let's say I have gene A. So I'm going to design the probe. Probe is basically the complementary sequence for this particular gene. So I have the probe, and now what we are going to do is I'm going to load the sample in this particular area. So what we have is, let's say each well is there. So we are trying to load the sample. So for example, if I try to explain for this particular part, that is the green one. In this case, let's say I have loaded the sample from healthy cells. I have loaded the sample from pathological cells. Then I try to load the probe for gene A. Okay, I have loaded. Now, if I'm getting the green signal, it means this probe is getting hybridized with this particular transcript. Okay, then it is producing the green signals. if whenever the hybridization is going to take place, they're going to produce the signal. If this particular transcript is involved, they're going to produce green signal. If these transcripts are involved, they're going to produce red signal. So depending upon signal, we can figure out which sample has the expression profile for this particular gene. Because if they have the expression profile, they're going to produce the gene, and gene will have the transcript part, and we already have the complementary sequence, so they're going to bind it and give me the signal. So we are getting the green signal. It means the normal cell has the expression profile for gene A, and pathological cell do not have the expression profile for gene A. It means gene A will may be present, but this is in the inactive state. Means this is not producing any transcript. In this case, they are producing the transcript. Same thing, if I have to check for other genes, we are going to load in each cell. If we are getting yellow signal, it means that particular gene is active in both cell, means both healthy condition and the pathological condition. If we have the red, it means it is only expressed in the pathological cells, and normal cells is not showing any expression. And the gray part is neither the healthy cells nor the pathological cell has the expression profile for that particular gene that we are checking. So this is the way we try to do the comparative analysis, and this is the way we try to do the interpretation part from the coloring pattern. So is it clear, or anyone has any doubt?
It's clear.
Okay.
But how it is the part of NGS? I didn't get like uh
So let's say, for example, we are using NGS in this particular location. So this is the traditional method to generate the gene expression data. So I'm explaining when we are going to encounter with gene expression data, basically the transcript or transcriptome study. So we have a
mhm.
lot of experiments. So this is the first experiment that was used, that is microarray.
Okay.
In this case, we are basically isolating all the RNA. We are adding the tag. We are designing the probe. It will bind. We will get to know this particular gene is present and showing activation, means they are producing transcript.
Mm-hmm.
If it is not giving signal, means that particular gene is inactive state. So this is the simple study. But in this case, we move to the next study, that is the RNA-seq part. So in this case, we are only limited for few genes, means we can do the study for only few genes. But if you want to do the study for the entire gene that is present in human, let's say we have more than 20K genes. So for that condition, we are going to go for NGS, where RNA-seq, where NGS is used. So we are going to isolate all the mRNA, going for the fragmentation, creating the complementary DNA, going for amplification, sending the sample for sequencing, whatever the reads we have, going for the alignment, and
Oh,
then
okay.
getting the expression profile.
Actually, that this diagram was not there, so I was
Yeah,
in
so
this.
that time
Yeah.
I was explaining the microarray part.
Oh yes. Fine.
So these are the two different techniques. In this
Mm.
case, we are limited to probes. In this case, we are not relying on the probe part. So we can have the expression profile for all the genes that is present in the sample. So we are doing the sequencing of all the RNA molecules that is present in the sample. That is the concept of RNA-seq. So this is the study we are going to do it. Means we are going to compare between two conditions, but we are not limited to only few genes. We are going to check the expression profile for all the genes, whether it is active or inactive. If it is active, whether they are showing more expression profile in normal compared with the treated condition. So these kind of comparative analysis comes under RNA-seq part.
So I have a question.
Yes.
So when you're working with a rare population, like for example, in my case, I am working on a population which is present in the lungs in the number of, let's say, maximum of 20,000 in a whole lung of the mice. So I am unable to get the RNA, and as like consequently I'm not able to get its transcriptomics done. So what do you suggest in that case? Because I cannot enrich those cells more than 50,000.
So what exactly the experiment? So you're doing to collect. So you're trying to do the first genetic connect, that's genetic information collection. So you're
So
going
yeah,
for isolation. Yes.
yeah. So what we do is like we just harvest the lung from the MTB infected mice after like two months of infection, and
Hmm.
then I sort those cells through flow cytometry.
Okay.
And the number that I get from a one mouse is like 20 to 25,000. This is maximum I'm telling.
So this is for cells.
This is for cells I'm talking about.
Yes. Yes.
And because the cell number is so less that I don't get the RNA, whether I do it in the lab or I give it to the company. So I've done this like thrice, but I'm unable to get this transcriptomics done for those samples.
So is it because you're trying to rupture the cell, but you're not able to capture it? That may be
Yeah,
the case.
so what I have done is, what we do, we harvest the, like we sort the cells, then we, through the trisol method, we isolate the RNA. But the quantity of the RNA is very less. It's just like 1 to 2 nanogram per microliter.
Yeah. So you can go for amplification.
No, it's not. It's not like it's very difficult to get into that amplification because it does not pass the QC. Now they say that you have such a less RNA, so it fails QC.
Okay, I'm not sure about the how exactly in the wet lab part because the logic is you have the RNA, you're going to convert it into complementary DNA, and then
So
you
I'm
can
unable to
increase the
get
amount.
cDNA in the lab. Also, when I am doing it myself, like without any fragmentation or anything, I'm unable to get cDNA because of the low quantity of the RNA.
Uh, but I'm still confused if whether, let's say for example I have only two RNA, let's say. So
Mm-hmm.
if I'm going to add the enzyme, it is going to do the reverse transcription.
Yeah. So the CT value, when I make the cDNA, the CT value that I get is 30, 32, because the reads are very less now.
So what exactly the others are doing? They are going. So for example, there are a lot of literature which is working on the same domain or let's say same model. So how exactly they are
So,
performing?
so what people have done in literature is that they have done single cell RNA-seq, but that is a very expensive technique. Every lab
Correct,
cannot do that.
correct, correct, correct.
But I have not able to get this transcriptomics data on any of the paper regarding those cells.
So maybe this is the reason that's why they have moved to single cell. I'm not sure, but if
Mhm.
no one has reported any of the capture part
I have
using
done
the,
the proteomics for that. The people could do proteomics, but not the transcriptomics.
but that is the valid confirmation. For example, anyway, if we have any information from the transcript level, so we are going for the
I know.
validation using proteomics
Yes,
also.
yes.
But if we have the confirmation for proteomics, so basically you have the result part. So you don't have to go for transcriptome part.
Actually, my one of my purpose was to do for this DTU and DEG expression.
Okay, so you want to see compare what exactly the expression profile between the condition, if you're going to
Yes.
give any drugs or not.
Right, right. So that was one of my objective to accomplish. But yeah, because of that, so that's why I was asking, is there any way to get that?
And no, I cannot recommend anything because I haven't done these experiments because I'm
Okay,
not from the wet lab part. So mostly
okay,
I've spent most of the time on the computational part.
okay,
But you
okay.
need to check, I guess, if someone has performed, so they generally add the method information and the entire thing. And you can also ask the author because you can write a mail to them if they have performed. So generally they provide, okay, we have used this solution, we have used this protocol. So they are
Mhm.
happy to share. If you're going to ask, this is not for publication, we just want to know or replicate the things to validate
Oh,
something.
yeah, yeah, yeah.
So you have to write in such a format so they can get to know, okay, they just want to study more, or let's say they want to replicate to see whatever the result I have published, that is correct or not.
Yeah. Okay, okay. Thank you.
Yeah. Yeah. So this is the part of RNA-seq. Now this is the last slide I'm going to cover. So because yeah, nowadays we have bulk, single cell, and the spatial. So as of now, we are going to cover this part in this entire workshop. But I just want to give a discrimination what exactly these methods are, so that you have a clear differentiation and you have a clear idea what exactly these methods and when we have to use these methods. Bulk RNA is simple. I can give an example. Let's say I'm going to prepare a juice where I have added a lot of ingredients. For example, I have added strawberry, I have added orange, I have added banana, but the quantity of strawberry I have added while making the juice is very high. Now I'm going to ask two questions with you. Like, could you please tell me, let's say this juice is made up of which fruits? That is the first thing. And the second thing is, could you also tell me the quantity I have added? So one answer you're going to provide me is this is the strawberry juice because you are getting the rest of strawberry. And because of the quantity I have added for strawberry is very high, that is kind of acting as a dominant character and masking the other features. Like because I have added apple also, banana also, but we are not getting the taste because the quantity is very less. Same
Mm.
thing is happening in the bulk RNAs. We have a tissue. We are rupturing the entire tissue to collect the genetic information. So we have the transcript, and basically we are providing the average expression profile. Means, for example, tissue is made up of cells. So cell is the basic unit of life. So let's say this entire tissue is made up of, let's say, 7 cells. I cannot say this transcript was coming from this cell or this cell or this cell. I can say this entire tissue is showing the expression profile for this gene. So that is the concept of bulk RNA. In case of single cell, we are not capturing the entire tissue. We are basically first isolating each cell. So now, let's say the entire tissue is made up of 8 cells. So we will have 8 cells. Now you will rupture each cells to collect the genetic information, and you will have the expression profile for that particular gene at a single cell level. Means now you can say what is happening within a cell. In this case, we are losing the information that transcript was coming from this region or this region or this region. But now we have the idea, okay, which cell is having the expression profile for gene A or gene B or gene C. So that is the case.
This is a reminder.
Alexa,
Dinner
stop.
time.
So this is the thing. So we have single cell part and we are trying to check the expression profile at a single cell level. But one drawback for the single cell part is, one advantage is we can have the expression profile at a single cell level. Drawback part is we are not keeping the information these cells are coming from which location. Means this cell is situated at this periphery region or the center region, where exactly we don't know the coordinate, x and y coordinates for this cell. It means I can say in this cell, we have a tumor sample, we have a cancer. Let's say, for example, this is the sample I'm handling. Let's say I'm handling the breast cancer data. I can say in this particular sample, there is a cancer cell, there is an immune cell, there is a fat cells, there is a stroma cell, let's say. But I cannot say how exactly or where exactly the tumor cells is located, where exactly the immune cell is located, whether they are in the close proximity, whether there is any infiltration of immune cells. I cannot resolve these kind of questions. So for that purpose, we have to go for spatial. Spatial is nothing but a single cell. We are doing the single cell, but also we are keeping the location. That is the spatial coordinate. If you see this image, I can see the strawberries present at the periphery, then we have the circle for the grapes, then we have for the oranges. So we know the location. So it means I can say this is the tissue. So the tumor cell was present in this region. This is the region where immune cell is present. This is the region where other cell is present. So I can figure out, I can say where exactly the cancer cell is present on the sample or on the tissue. That is, we can solve using spatial. So these are the differences from bulk, single, and the spatial part. Anyone has any question, any doubt?
No, it's good.
Yeah. Okay. So we have covered the first session, and
Sir?
yes,
Yeah, so in the last slide,
yes,
this is the name, this is the name of the technique, like bulk, the RNA technology, and like single cell RNA, like that.
yes. So these are the essay, or you can say the experiment or technology. Yes.
Okay. Okay. And how, like, which one is beneficial at which experiment? Can you just elaborate on that?
So as I have given an example, let's say I have to find out within my sample what kind of cell is present. So I cannot solve with bulk RNA because bulk
Okay,
RNA is
okay.
just giving the
Okay, fine.
comparison.
Yeah, yeah.
So for that part, I have to go for this part. But if I have a question, I need to see whether immune cell is present between the tumor cell or not. Is there any
Mm.
infiltration is happening? I cannot see those information with single cell. I have to go for spatial.
Okay, fine. And when the bulk is used?
When you have just simple, you want to see the expression profile is active or not within two conditions.
Means that particular means that particular expression is happening or not. That.
Yes, yes. Yeah, for example, I'm giving some drug to you and I want to see what is the effect on couple of genes that I want to study.
Mm.
So I don't have to spend much money on single cell because this is costly. This is most costly.
Okay, okay.
So I can solve the problem with the simple process. So I'll make my life complicated.
Okay.
So that's the concept.
Okay.
Yeah. So we have covered the session one, and yes. So one thing I just want to add, I forgot to add in the starting part is you will get the recorded lectures for each session. In the let's say for example tomorrow morning you will get the lecture. Same way you will have the access for all the recorded lectures next morning. Second thing is you need to use your Gmail that you have given to access the recorded lecture.
Mm.
Third part is, let's say, for example, if you have still any doubt, let's say, for example, you're going to explore some research papers or some open resource. If you have any doubt, feel free to come up with your doubt or question, and we can discuss in the next session also in the starting 5 to 10 minutes.
We can do one thing, like as I'm a fresher, like very like I'm just starting this. I don't have much idea about bioinformatics and all that. So
Okay.
just can you just provide means before every lecture, can you just in the morning itself, can you just say uh, s- uh can you just provide some material that I can go through before the lecture, so just I
Yeah,
can have some idea
I can
or some
provide.
topics.
Correct. I can provide, but as of now, the topics we are covering, so it's very hard to cover the topic within a single day. What are the resources I'm going to provide? But still, you can try. I can provide all the resources that I'm going to cover.
Mm.
You can try to explore one by one. And whenever you have a time, you can try to read those parts. Yeah, that I can do.
Okay, okay, fine.
Yeah, also remind me once, let's say, for example, in the afternoon, if I'm not able to provide the resources, let's say till 11 or 12, so just remind me once on the WhatsApp group because I have some other work. So sometimes I miss those information or I just skip my mind. So please remind me once.
So the anything which is basic to this field, you can also provide now itself if there are some basic papers or anything.
Yeah, I need to check my document. So I need to go and look for the document that I have created a long back. So I need to just collect the links part to provide it on the WhatsApp group. So it will take some
Okay.
time.
Sure, sure, fine.
Huh. And the
Okay.
last point I just want to add is after the session, you have to give the feedback. Uh, So like whatever comfortable to you, for example, either in the format of audio, video, or text format. So that will be helpful.
Sure.
Sure.
So I have
Yeah.
a question. So this
Yeah.
entire workshop will involve the coding and everything, or it will be like how it's planned?
So let's say, for example, I'll be honest. So we cannot complete everything within 7 days. For example, let's say whatever you're going to learn, it will take, for example, 4 months, 5 months, or 6 months. And
Mm-hmm.
whatever the expertise I have, I cannot teach everything.
Right.
So I'll try to cover the basics so that you will have some idea, so that you can also start exploring and reading the stuff, and you can do the analysis. So I just gave in some idea. Okay, this is the feature, this is the workflow, this is the step, and this is the parameter, this is the code part we generally write. For example, if I'm going to explain the QC part, I cannot explain all the QC that we use currently with the data, real data. I can
Mm-hmm.
explain few parameter. Okay, this is in the QC part, this is the read filter, let's say this is the other filter, and this is the value we are going to put, and this is the range we can select. Even the value can change for your data. Let's say, for example, I'm handling breast cancer data and I'm using a threshold, let's say 50. If you're handling other data set or different models, the value will change. So you are not going to say, okay, you told me you need to use 50, but I'm using 50, I'm not getting the correct result. That's not like that. You have to do a trial and error part. You have to repeat the workflow with different parameters, and you have to do the interpretation part, whether this is correct or not. Because you are doing the project, you have the biological concept, what exactly you are expecting from the data.
I have one doubt.
Yes.
So as you said that it will not be completed in 7 days, it will take more time. So you will take more time. You're just saying that we have to
No,
try.
no. I'm saying no one can provide the resources or the coverage part for the entire RNA-seq within a 7 days or within a 1 month, I can say.
Okay.
Huh. So let's say if you are, I'm saying as a friend, for example, if you're going to join any workshop, let's say they are saying, okay, we can cover the entire protein sequencing analysis, whether it's RNA-seq analysis, single cell analysis within 20 days. So they
Mhm.
try to touch the concept only
Yeah.
to give you some idea what exactly this domain is, what exactly people are doing in this domain, how exactly people are doing their data analysis part. So same thing, I'll try to do it. I'll try to use some dummy data to explain the workflow, how you're going to start with the FASTQ file, how you're going to do the alignment, how you're going to do the quantification. But if you are expecting, I can explain each point in the alignment part or each parameter in the alignment part, that is not possible. That is ideal case.
Okay. And also it was provided in the workshop that we'll be doing means we'll be handling the data. So is it
Yeah,
going
so
to happen?
you are going to replicate the result that I'm going to use. For example, whatever the data I'm going to use, I'm going to provide the link and you can try it out.
Okay. Fine.
It means, for example, today I'm writing 5 lines of code. You need to practice the same code at your home after the session with the same data.
Mm-hmm.
So that's kind of hands-on experience.
Okay. Okay.
And depending upon your interest and requirement, you are going to explore more.
And after the workshop, also we'll be having a lot of doubt because obviously we are just starting it. So you'll be there to
So let's
monitor
say
that.
let's say for example, you can ping me anytime on WhatsApp group. Whenever I'll be free, I'll be happy to resolve your question. But I cannot guarantee. Let's say for example, you're going to say, "Hey, Asis, I have some doubt. I have some question. So could you please resolve this issue? Can we connect?" So no. For example, let's say generally I have free time on weekend. So for example,
Okay.
you're going to post a message on Monday. I'll say,
Mm-hmm.
okay, I'll provide the solution or I'll provide the resources on, let's say, Friday or Saturday morning.
Mm-hmm.
So till time you have to wait. It's not like that you're going to provide us the question and you are expecting me to resolve it quickly. So that is not the case.
Okay.